So now turning to our speakers, and it's plural, we have two speakers today and a special activity. So I'm going to introduce both of them at the beginning. First up will be Tim Martinson, and I've known Tim for decades, actually. He's a member of the Executive Council of CAPE, so we have one of our own speaking today. He's a Senior Extension Associate Emeritus with the Horticulture Section, the School of Integrated Plant Science, or SIPS, and he was based at Cornell Agritech in Geneva, New York. He has spent much of his career as a viticulture statewide extension specialist, and Tim retired in 2022. And right after he retired, he was interviewed, and the question he was asked one question was, what was your biggest surprise when you started an extension with the Finger Lakes Grape Program? That was after working for several years as a research associate. I don't know if Tim even remembers his answer, but he says his first thought was, what am I doing here? And I think a lot of Cornell undergraduates have that exact same thought. But Tim quickly figured out what he was doing there and found out what was needed. And he spent 25 years doing outreach and applied research to support the growth and profitability of the wine and grape industry in New York State. So that means in extension, he worked closely with faculty, regional extension educators, and the industry. And he provided growers and wineries with information to help them with management and production decisions. And I'd like to share one fun fact about each of our speakers. Tim recently joined the board of the Cayuga Lake Watershed Network. And I know this because my husband, David Weinstein, in the audience also joined the board at the same time. And since the acronym for this network is CLWN, we call it Clown at our house. So in his retirement, Tim has chosen to join the circus. Our second speaker will be Bruce Reisch, and he's a professor emeritus and also in the section of horticulture with SIPS, and also was based at Cornell Agritech in Geneva. And he retired in 2023. And Bruce specialized in the development of new grape varieties and also grape breeding techniques. And in his 43 years at Cornell, his team released 14 new grape varieties. 10 of these were wine grapes and 4 of these were seedless table grapes. His field of study has taken him to France, Italy, Germany, Portugal, Israel, Hungary, Turkey, Thailand, Switzerland, China, and Japan. And Bruce, I don't know if you were doing grape research or if you were in training to be the Secretary General of the UN with that amount of travel. And here's a fun fact about Bruce, which he doesn't even know. Bruce and I were at the University of Wisconsin in Madison at the same time in 1980. He was finishing his PhD in plant breeding and genetics, and I was starting my career as a statewide extension nutrition specialist. And with the 40,000 people on campus, I cannot believe I didn't meet him. So I'm standing between you and a taste of wine. So let's first welcome Tim, and he's going to give us instructions about that activity and also help us learn why it's time to embrace hybrids, how new wine grape varieties will lead to a more sustainable wine industry. Welcome, Tim.
Thanks, Christina, and it's a real pleasure to be here with my esteemed colleague, Bruce Reisch, with whom I've worked for many years. And but I'd like to start out with asking you a question. So show of hands, when you go to a wine store, what do you gravitate toward? Is it something called Chardonnay? Raise your hands. Okay. Is it something called Cabernet Sauvignon? Okay, a lot more hands there. Pinot Noir? Okay, a couple Pinophiles here. Is it something called Marechal Foch? I'm not seeing any hands. How about Traminette? There's one, Bruce. You're a cheater. Okay, so the point of the matter is that when you're selling wine, the name of the variety is important. That's kind of unique in the commodity world, except for maybe apples where, it's Honeycrisp, but it has consequences for how people accept new varieties. So I just wanted to make that point and at the start here, and then think about that as I proceed with my talk. So about the wine tasting, how many of you have wine? Okay, almost everybody. So what I would like you to do is to taste these two wines carefully as I complete my talk, and then decide which one of them you prefer, and decide which one of them you think is the hybrid, and which one of them you think is the vinifera grape. Okay, very good. So the topic today is sustainability and grape breeding. Grape breeding and genetics is kind of complicated, very long term, but sustainability is something that our industry and wine grape industry has been very interested in for a couple of decades now. So everybody wants to be known as an ecological farmer. It helps with marketing your product. And in addition to sustainable, there's several other labels that have come along that people like to use when they're selling their products. Biodynamic, organic, the latest being regenerative. So this is something that is a continuing theme, but it's something that our growers have been interested in for a while. And then the other topic that is grape breeding, and I want to explain why these grapes may be more sustainable than the grapes that you all mentioned when you go to the liquor store. And that have a more prominent name. Okay, so first, sustainable viticulture. What's it all about? Well, in the early 2000s, we were asked by the industry to develop a workbook that would be used by growers to decide whether their practices were sustainable, what things they might want to change, what things they might want to keep the same. So what is this all about? Well, it involved us putting 107 questions, more or less, about their production practices, the whole range of things from soil erosion, pest management, floor management, nutrient management, etc., biodiversity, continuing education in this, and then they would sort of score themselves on a range from 1 to 4, depending on how sustainable the practice was. For a long time, that was just a workbook that people used. But now, as of 2023, there's a formal certification process that's sponsored by the New York Wine and Grape Foundation, which includes a trademark and, well, they call it a trust mark, and a lot of marketing information so that people know about it. And that's part of the point of sustainable viticulture, is you can do these practices quietly, not tell anybody about it, but the point is that you want people, including your neighbors and your customers, to know about it so that they will know that you're trying to practice ecological agriculture. So some examples, it's kind of complicated. There's a lot of things because growers do a lot of things, but here's a couple of examples. So back in the 80s, people used to run their discs through the vineyard 4 times. So now they've abandoned tillage. So no tillage, permanent cover crop. That's good for soil health. A lot of them, again, in the mid 80s, started rolling out big bales of straw in their row middles. That helps with moisture retention, adds organic matter and so forth. And this is surprising to me because at least among the people that grow vinifera, almost all of them that I've talked to have stopped applying any nitrogen, any supplemental nitrogen at all. Why is that? Because if they put any on, our soils have the ability to supply enough, and the vine growth goes wild and crazy. The other thing in the Finger Lakes is if you have your soil pH right, you don't have to add any phosphorus. So this ties in with biodiversity and pollution, because there's no way with these stats that we're contributing to phosphorus loading in the lake. Okay. And the other thing is, well, if you go out to California to the Central Valley, you'll see a sea of grapes, no other plants. But in the Finger Lakes, even the largest vineyards have a lot of biodiversity around them. So that's another thing that we ask questions about. On the other side, and Bruce is going to talk about this, is the fact that whole genome DNA sequencing, what I call cheap DNA sequencing, has revolutionized grape breeding. So when Bruce started, the parents were chosen basically by field evaluations. Nothing more complicated than saying, well, I like this one and these traits. I like this other one and those traits. Let's see what happens when we put them together. And surprisingly to me, there's very little knowledge about how traits were inherited in grapevines. So it was very much an empirical process. So starting in 2000 and continuing on, they had more and more elaborate genetic sequencing platforms, culminating in 2016 when I was involved, this platform called RAMP-seq that has 1,600 DNA markers and it covers the entire grape genome. So this is a tool that they have that they can use to figure out which traits are associated with which chromosome. Bruce will talk about that a little later. So I want to go through the varieties in New York, or in the Finger Lakes in particular, and how they got here. So surprisingly, and this report just came out last week, they count 93 varieties of wine grapes grown in the Finger Lakes. That's way, way too many. But the top 30 in this list, and this is only a portion of the acreage where growers responded, comprise 92% of the acreage. And we divide them into 3 different categories. Ones we call natives or Labrusca types, some we call hybrids, and some that are Vitis vinifera, the classic wine grapes that you all know about. So I want to go through when these 3 successive waves of introduction into the industry. So starting in 1865, since 1865, we've grown Native American grapes, Concord, Niagara, Catawba. And these, I think Lake Country Red is something that it's a Concord-based wine that typifies what our industry was known for. Lake Country Red, Lake Country White, all based on Labrusca type grapes. So after World War II then, the French hybrids were introduced into our industry. So those that introduced them hoped that they could get a different range of flavors than what they were able to get with the Labrusca type grapes. And there's several of them, and several of them got their names here in the Finger Lakes. So we named some of these varieties. So why did the French start hybridizing grapes? It was in reaction to a big crisis. Phylloxera became an invasive pest in France and started killing their grapevines. So that's what inspired people to start using North American germplasm that was resistant to the phylloxera, rating roots to see how many phylloxera colonized them, and trying to combine the wine quality that they liked with the phylloxera resistance. So they're called in France hybrid direct producers. We call them French hybrids. But that worked, and they were widely planted across France. But then this other solution sort of came to dominate things, and that is grafting. So they found that if you put an American rootstock based on American species that was tolerant to phylloxera, you could then grow your own varieties that you've known and loved for so many years. So that took over, and the government of France started to ban planting hybrids. Why? Because they say inferior wine quality. And this is something that I call the hybrid taint, okay? Hybrid grapes have been thought of as second-class citizens, and this is what we produced through the mid-80s, with the large wine companies and so forth. But how did people characterize them? Sweet, foxy, grapey, simple, jug wines, okay? So we weren't a serious wine reason to the wine critics and wine aficionados at that point. Okay, just a little sidestep here. Cornell hybrids that Bruce is going to talk about. They've been breeding for a long while. I believe Cayuga White was, the cross was made in 1948, but released in 1972 as the first wine grape released by Cornell, and then spanning on to Airville in 2023. So these are grapes and different flavors that don't fit that hybrid stereotype that people have. But it still exists, believe me. So in the mid 80s also, again, people have been growing them since about 1960 with Constantine Frank and so forth. But it was really in the mid 80s that these took off. And so we had, became known for Riesling and Cabernet Franc, and we're able to grow vinifera varieties here, largely because there's new fungicides that helped us control the diseases, which I'll get into in a minute. So what happened was that these vinifera grapes and Chardonnay, Riesling, Cabernet Franc, to a certain extent Pinot Noir, elevated the perception of New York wines. This was a kind of a gradual process, but as of as of now, the Wine Spectator has said, yeah, we're a real wine region. They make quality wines. This is touting two Finger Lakes Rieslings that got 93 points on their scale, which means they're exceptional wines. So if you talk to growers now, if they're planting grapes, it's probably vinifera, okay? They're probably not planting many hybrids or Labrusca types. So this is great, but what's the problem? Well, there's two classic problems, two weaknesses for vinifera. The first is winter injury. This is showing a vineyard after the winter of 2003, in which there was a sudden drop of in temperature, sub-zero temperatures, and that killed about a third of our vinifera grapes. And the remaining ones, we had to do trunk replacement and all sorts of things. So they're not very winter hardy, which is a problem in our region. The other one is disease susceptibility. They're highly susceptible to a lot of diseases. And I'll get into that a little bit more in a moment. So this makes them expensive to produce. If your vine dies, you have to wait 3 years before you get more fruit, etc. If you have to, spend a lot of money on fungicides, that's a big cost, and so forth. So this is an article that appeared in the New York Times in 2011. Here's the title: "Lack of Sex Among Grapes Tangles a Family Vine." And it's all about the fact that when they started looking at the genetics with DNA fingerprinting, so to speak, they found that a lot of the most popular wine grape varieties in the world were very closely related. A famous example of this is Cabernet Sauvignon, which is the child of Sauvignon Blanc and Cabernet Franc. So that's a very narrow sort of genetic thing, even among the vinifera. The classic ones tend to come from a narrow genetic base. And so I wrote this article called "Frozen Genetics of International Wine Cultivars," because there's sort of 10 or 20 that dominate the world, and they're all come from the Middle Ages. So if you had Cabernet Sauvignon in 1700, it's the same vine. It's genetically the same. So Napoleon would recognize Cabernet Sauvignon if you tasted it today, right? They're all vegetatively propagated, so that means that there's no genetic recombination, so forth. And their big weaknesses, as I mentioned, winter hardiness and susceptibility to diseases from the New World. So if they originated in the 1600s, well, they had these invasive pests come into Europe in the 1800s, the mid 1800s, not just grape phylloxera, which I already mentioned, but powdery mildew, downy mildew, black rot. These did not exist in Europe, and the vines that are grown there did not co-evolve with these pathogens, okay? So they're highly susceptible to all of these things. So what's the consequence? Well, if you want to grow Chardonnay in the Finger Lakes, you have to control 5 different foliar fungal diseases: powdery mildew, botrytis, downy mildew, Phomopsis, black rot. And you're probably putting on 13, 15 fungicide applications. Okay, so that's great. Well, out in California, they have a drier climate, and they don't have some of the diseases. All they have is powdery mildew and botrytis, no black rot, no downy mildew, but they're still putting on 6 to 10 fungicide applications. And this is true whether they're conventional growers, organic, biodynamic, or regenerative. And most of it's sulfur because that's organically approved, but they still have to put on these fungicides to produce saleable grapes and wine. So here's my question. Can the industry really claim to be sustainable when the grapevines require 10 to 15 fungicide sprays? For me, it's a big blind spot in certification programs, is that they sort of soft pedal this intense amount of inputs they have to do. And moreover, the fungicides don't always work. The pathogens have short generation times. They develop resistance very quickly. I remember this fungicide called Abound that lasted 3 years in our industry. It was the miracle drug when it came out. It controlled all of the diseases, but within 3 years, powdery mildew was resistant to it, and it was ineffective. So there's no guarantee that we're going to be able to even use these in the future, or that there's going to be replacements. And just to put a fine point on it, this is what grapes look like if you don't spray them, or vinifer grapes in the finger. This is the unsprayed check in the disease management trial. And you can see it's defoliated, and there's all sorts of nasty looking clusters there. So that brings me to genetics. So people are not seriously considering this out in California, but genetics might provide a great solution. Disease resistance and cold hardiness come from these Native American species, these wild grapevine species, and they can be incorporated into new varieties. And here's a sort of another fact. If we look at the evolutionary tree, Europe has 1 species of grapevine, but Asia and North America have about 30 to 50 of them. So there's a lot of untapped genetic diversity, including lots of disease resistance because of 5 million years of coevolution with North American grape. So this is a resource that needs to be tapped for to make more sustainable grapes. And that's what Bruce is doing. So, but they're also doing this elsewhere. And French researchers released resistant varieties in 2018. So this is a big turnabout in which they're starting to think about hybrids, and they're starting to do it because they, with 8% of the land area, they apply 20% of the pesticides in Europe. So what can this do? Well, comparing these two with conventional varieties, they went from 15 sprays in a conventional program down to 2. So that's a 86% reduction in inputs that they had. And I'm close to the end here. So really, what grape breeding offers is more sustainable disease-resistant varieties. They can stack powdery mildew and downy mildew resistance genes into one vine. You can drastically reduce the number of fungicide sprays. And this translates into fewer tractor passes, less soil compaction, lower greenhouse gas emissions, less embedded carbon from manufacture of all these inputs that are needed, and a reduced cost of inputs. So this is why I say it's time to embrace hybrids. Now, have you all tasted their wine? Okay, I'll get there in a moment. But the last thing I want to do is to talk about Aravel, Bruce's latest release. I guess your final release, Bruce. So this was a cross between Riesling and Cayuga White. And what are its advantages over Riesling? Well, Riesling rots, so they can't grow it in the central, in Virginia or the mid-Atlantic area because it's hot and humid and the grapes rot. Aravel does not rot. So what's the story behind this? Well, Bruce made the cross in 1981. These growers out on Bluff Point and Keuka Lake planted the grapes in 2010. This wine, which is actually a varietal wine called Heart of the Lakes, was started to be made in 2020. And Bruce finally named it in 2023. So that's a 42-year thing between the cross and the release and naming. So very long-term stuff. I don't know how you do it, Bruce, you know, thinking so long-term, but that's the fact of life with grapes. Okay, so the wine test, taste test. I'm going to ask you a couple of questions now. Okay, how many, first is about preference. So how many of you preferred wine number one? Let's have a show of hands. Okay, that's about half, okay. How many preferred wine number two? That's got to be everybody else, right? Okay, it looks like it's split about 50-50. Okay, how many think wine number one was the Riesling? One, okay, there's 7, 8 of you. How many think wine number two is the Riesling? That'd be the rest of you. Okay. Well, we'll do the reveal at the end of Bruce's talk. So I'm gonna, I'm not gonna reveal it now. So I'd like to turn it over to my colleague, Bruce.
Okay, so yeah, this makes sure that the audience stays in place right till the very end, till the reveal. So yeah, it's nice to be at Kendall and to see some of my old friends. It's really nice to be here. Thanks so much. As Tim said, I'm going to give you a little rundown on what happens in grape breeding and why we can now do grape breeding with a much greater deal of efficiency in the process. Made possible by advances in genomics and genetics. When I started in 1980, we would choose parents, as Tim said, based on complementary traits. This parent had traits A, B, and C. Another parent had traits D, E, F, X, Y, and Z. We wished to combine those traits in a new variety. So we'd make the cross and evaluate in the field and evaluate the wines. So every seedling is different, just like you're different from your brother or your sister. In the world of grapes, every individual progeny of two parents is different. And that's been a pretty successful formula from the late 1800s to present. There are a lot of good varieties that have been produced that way. But you know, it's challenging, and it can be less challenging if you have the right tools. So in 1980, and even today, these were some of our challenges. You need many vines to breed grapes. You only need one good vine. As Tim mentioned, grapes are vegetatively propagated. So one good vine gets propagated into many vines to plant vineyards. Riesling comes from a single seedling from centuries ago. Cabernet Sauvignon from late 1700s, a single seedling gave rise to Cabernet Sauvignon, and then cuttings are taken. You have the need for year-round vineyard operations, nursery operations, greenhouse operations. It's expensive. You have a 3 to 4 year generation cycle from seed to flowers. So it takes time to make a cycle of crosses and then to make crosses with the next generation of grapes. And you require multiple years of evaluation, because if I were to tell a grower, Tim would know, I tell a grower I have a new variety and I have one great year worth of data, they're going to laugh at me, right? uh, Growers are about to make a 30-plus-year investment. They don't want 1 year of data. They want to know, how did this grape do for 5 or 10 years, and did it do well in the southern Finger Lakes, northern Finger Lakes, this soil type, that soil type. So you have some challenges. It takes a long time. Under ideal conditions, maybe you can go through seedling trials and propagated vine trials and multi-location trials in a 20-year period. Often it takes much more, as in the case with Aravel. So I looked around in the 1980s. What can I do to really have an impact over the course of my career? There were a lot of grape varieties out there. How can I improve on what's out there? First of all, I know this need to get away from spraying. I know that wild vines grow without being sprayed, and you hardly see disease on wild vines, whether they're in the Finger Lakes or in Pennsylvania or Illinois. So one of the first things I did, when I thought about where my impact could be, is I said, well, can we breed disease-resistant varieties that would be the foundation for integrated pest management, for sustainability? Looked at what was growing in the vineyards, and we found some types that were neglected parts of the vineyards I had inherited, and they were as clean as possible at the end of the growing season. This was one example, this hybrid of rupestris and cinerea, and this formed, this we later used in crossing. And I had others that I found as well in the program that were quite disease resistant. So we felt, hey, we can do some things with this. And I always felt that we needed more genetics, more knowledge of genetics, but sorry, let's go back. Starting with what I had in the 1980s, I knew I can go out into the field, make crosses, and screen these grapes basically under no spray conditions and save the very best. So this was an efficiency that was within reach in the 1980s. We can eliminate 80% of the progeny, the most disease susceptible, and save the top 20%. And now my vineyards are much smaller, and I can focus on the best material possible at that time. I had no idea what genes they had. So my point here is that many of the releases that I put onto the market were due to crosses made by my predecessors. And I was very lucky to inherit some really good material. But Aramella, for instance, was from a cross made in 1976. Arundel was one of my crosses. Hey, by 2013, a cross I made in 1995 was released. So there's Arundel out there in Virginia, New York, Kansas, and it's doing pretty well. So Arundel is out there, NY81 is out there, Everest Seedless is one of my crosses. So yeah, it takes a while. You have to be patient, I guess, is the answer to be a grape breeder, and have a little mind for numbers, because all of these have numbers, not names in the program. But there was success based on the old technology. So what could we do better? I always felt we needed genetic maps of grapes. We needed maps to be able to locate genes of importance. And early in my career in the 80s and 90s, I was initiating studies in genetic markers that could help create maps of grapevine chromosomes. And we had some of the early publications in this area with older technology, but we could team up with a colleague. Norm Whedon was one of my key colleagues here in the early going. He left to go to Montana State, but came back recently for a visit, so I snagged this picture of Norm. And we had one of the early publications on what we call marker-assisted selection for resistance in grapes, resistance to powdery mildew. So we we're at that stage where we were starting to implement this technology, marker-assisted selection. What is it? It's using DNA-based markers, easy to assay markers, to tag genes of interest and to know whether that gene exists or not with basically forensic DNA technology. Does it exist in seedling 1, 2, 3, 4, 5? Which ones have the RUN1 gene for powdery mildew resistance? Which ones have the REN4 gene? When they're in the parents, well, the technology tells us which genes have, which seedlings have these genes, one or both genes, and which ones would be susceptible. And the technology has become so good that it correlates beautifully with what we see in the field or what we test in the lab. So we can just do the DNA test on every seedling that comes along, and we could stack multiple genes, RUN1 plus REN4 plus REN3. We can stack 3, 4, or more genes for powdery mildew resistance in one seedling. We can do the same thing with downy mildew resistance. It becomes an assay you can do with seedlings. It's more accurate than field or lab evaluations. It can be done with traits that are difficult to evaluate or require specialized equipment. It can be done as early screening for fruit traits. We can predict flower types, and that's really important in grape breeding. And it's several years before we see flowers or fruit. So we can predict the color of the fruit, some aspects of fruit quality. So this has become entrenched in the grape breeding process. Over time, initially when we discovered REN2 in the early going, a gene from that Illinois 547-1 grape that resulted in the publication of marker-assisted selection, there weren't many genes out there. We couldn't do a lot of marker-assisted selection, but there were a few in the early going. And then more recently, things have accelerated. So we have a lot of different genes available in grapes for powdery mildew resistance, many for downy mildew resistance, and a lot of other traits have been studied, and we have the inheritance information now. And Tim, you showed a slide of what happens in Katie Gold's program when you don't spray. And this is what happens in my vineyards when you don't spray. So you take an unselected population, you don't spray it, it looks pretty bad. You go to the next row, population that's undergone marker-assisted selection. Let's see, it's undergone marker-assisted selection. These are individual seedlings that have the RUN1 RPV1 genes for powdery and downy mildew resistance. And here's the whole row. These are adjacent rows. So there's a big difference. When you deploy the genetics, the vines survive and they thrive. In 2011, I had the opportunity to co-lead the VitusGen projects with my colleague Lance Cadel-Davidson from USDA in Geneva. And Tim Martinson was a part of the VitusGen projects as well. And working with all the grape breeders in the country, we had the opportunity to pull together computational biologists and people specialized in genomics and all the grape breeders and plant pathologists working together across the country under the USDA funding, Specialty Crops Research Initiative. $11 million came our way in this period of time to pull together 25 investigators, 10 different institutions, people working on wine grapes, seedless grapes, and apply technological innovations in phenotyping, the evaluation and genotyping, the genetics and genomics to deliver breeding lines with resistance to powdery mildew and high fruit quality. And one of these key advances, what Tim mentioned, was RAMP-seq, based on genome sequences in 10 different varieties and species shown here, and looking at the commonality of those genome sequences and developing markers only in the sequences, about 8% of the sequence that's common among all of these. So what's the importance of that? That makes the marker transferable. It makes it well anchored. It's transferable. If it works for me in my program, it should work for um, Dr. A and somebody in France, somebody markers were developed. The other thing that was important about it, we have something in genetics called crossing over. It separates markers from adjacent sections of DNA. So we need markers that are really close to the gene of interest. Early maps in the 2000s were pretty thin with simple sequence repeat markers. And then along comes genotyping by sequencing. And the maps are much more dense. There's much less space between the markers. You're more likely to find some markers close to genes of interest. And then with the RAMP-seq plus the GBS markers, really nice density to the maps and very low likelihood that the markers separate from the gene of interest. They should co-segregate with the gene of interest. They should really be top-notch predictors. So the VitusGen projects put marker-assisted selection tools in the hands of grape breeding programs around the country. We can go from thousands of seedlings down to hundreds, down to the hundreds that we want initially, the ones with powdery mildew and downy mildew resistance and good fruit quality and the right flower types and so forth. So it's resulted in numerous selections in the program. They're now fruiting after the VitusGen 1, VitusGen 2 projects. They're being tested in the program. Some of them have fairly promising wine quality. The VitusGen projects are also discovering new genes. Here are some 3 new genes were discovered from Vitis amurensis, a grape species from northern parts of Asia, a cold-tolerant grape species. So 3 new genes here for downy mildew resistance. 1 new gene from an American species here, Vitis X doaniana. It's actually a hybrid species, mustangensis by something else. So RPV33 was discovered. I've taken the initiative to try to incorporate these new genes into the program, leave them for my successor, just like my predecessor left some really good material for me. I'm leaving some, I hope, really good material for Matty Oravic, the new grape breeder at Cornell. From some of the crosses we made in 2006, one that might be a candidate to release in the fairly near future. It will be up to Matty, but this one has made good wine for several years and grows really well under low spray conditions, 1 or 2 spray per year conditions. So we're leaving Matty with material with newly identified genes for disease resistance, midwinter hardiness, slower deacclimation, material with a lot of vinifera germplasm in the background, some hybrids with Pinot Gris, Tokai, Merlot, Saperavi, Albarino, and really nice set of diverse material based on germplasm exchanges with a grape breeder in Germany. So let's see. Yeah, so we're going to wrap up pretty much on time. Here are the points that Tim and I wanted to make today. Just to summarize, the European grapes, your Pinot Noir, Cabernet, Chardonnay grapes cannot really be grown sustainably. Your hybrid varieties offer a great deal of genetic diversity compared to the vinifera grapes, and they can be grown sustainably. And I would say to you that many hybrids can make excellent wines. I think the success of Traminette, that's now pretty widespread in New York and other states, speaks to that. It's a Gewürztraminer hybrid that makes Gewürztraminer-like wines. So hybrids can be grown sustainably. New technology can be employed or is being employed and deployed to facilitate variety development. Sustainable hybrids are being developed not just in New York, not just in the United States in programs in New York, California, Minnesota, Missouri, and elsewhere. But sustainable hybrids are being developed in Europe, in Japan, in Korea, even my colleagues in France and Italy and Germany are really at the forefront of new variety development. And their world is opening up with fewer restrictions in the European community on what can be grown and where can it be grown. And I'd also say to you that new consumers are more willing to try something new. Newer consumers who are looking at blue wines and don't mind screw cap bottles, which actually help preserve the wine a little bit better. But new consumers are often looking for, something new on the market. They're not just looking for the same old Pinot Noir or Cabernet Sauvignon. The time is right, I think, to get these on the market. With that, my next slide reveals to you what you just tasted. Are you ready? Drum roll. Anybody? Okay. You remember what you said was vinifera, which one you said was hybrid? Some of you were right. Some of you were not. Nice going, everyone. Aravel was wine number 1. Cayuga White by Riesling. And for years, our data said Aravel makes a wine very similar to Riesling. And it doesn't rot. Well, I'd say actually it can get a little bit of rot. It's not immune to botrytis sponge rot. And it can be grown with many fewer sprays than Riesling. It doesn't have the newer genetics in it. It had genetics in it from 1980 when I started in the program, 80, 81 when I made the cross. But wine number 1 was Aravel, wine number 2, Riesling, both made by Weiss Vineyards. They're both 2023 samples, same year. Similar in residual sugar. So yeah, that can influence you if you're tasting one that's a little sweeter, one that's a little drier. A lot of times an audience will like the one with a little more sugar, though some of you might prefer dry. The data shows people will taste dry and sweeter, and most would prefer the one that's slightly sweeter. I'm sorry for those of you on Zoom, you're not able to participate in this fun exercise. But this audience seems to be very pleased up here. Next time, come in person. So I'm glad you're there. So at this point, I think Tim and I would open it up for questions. We have a little time.
We have 2 questions on the Zoom right now, and then we'll open it up. It says, why will hybrids remain disease resistant? Won't the diseases evolve and become a problem sometime down the road?
Yeah, that's a really great question. We're worried about that too, because there are documented examples of where certain diseases have overcome resistance genes, and those resistance genes are no longer useful. And that's why I've evolved my program from being a no-spray test program in the field to being a 2-spray program. And Maddie has continued that. We're working closely with plant pathologists to understand what are the effects of stacking multiple genes. And we do know that some genes give us resistance to certain races of powdery mildew that other genes do not. So certain genes can protect other genes. And an occasional spray, like a 2-spray program, well, and we need to know better, is it 2 sprays or 3 sprays and what the timing would be, but combining it with the most innocuous sprays possible will help us preserve the integrity of those genes. But we are working on that with plant pathologists because that's a really important question.
Thank
Can I
you.
add something to that?
Yeah.
Yeah, I just want to add that, the thing about the disease resistance that's in the plant is that it's always on, unless it's tissue specific. And And a fungicide is there for a few weeks and then it goes away. But if it's novel, then it can knock out the ones that are resistant to the plant itself. And so it provides just another way of ensuring that those resistant type pathogens don't proliferate and dominate.
Thank you. We have one more. When you talk about new hybrid grapes, are these made by classical crossbreeding methods or by genetic engineering?
All of the discussion we've had in this room today has been about classical hybridization. I have done some research on genetic engineering, trying to develop with disease resistance, but I didn't cover that in today's lecture. So all of the work on marker-assisted selection pertains to classical hybridization.
Beautiful. So you didn't cover perhaps the newer techniques like being able to do transgenic analysis by using Cas9 CRISPR techniques to introduce or to engineer. Is that not popular because people are so down on genetic engineered foods of any kind? Or and then the second question is that I come from the animal part of studies genetics, but and plants are notorious for polyploidy complex genomes. How many chromosomes are we talking about in grapes?
Okay, go ahead. The grapes we're talking about here in this talk today have 38, and the RUN1 gene came from the muscadine, which has 20 pairs or 40 chromosomes. So we have an ancestral hexaploid genome in grapes. It's highly diplodized after descending for millions of years from an ancestral hexaploid type of genome. We have 38 chromosomes
Great.
that we work with. And my Everest seedless, that was a little footnote in this talk, is a tetraploid variety with 76 chromosomes.
That's okay. So those
So-
are manageable numbers. So related to that is if you're isolating genes which are related to cold tolerance and disease, are you able, or have anybody tried to isolate those genes, which makes a Pinot, and a Cabernet? I mean, those subtle, given that it's
Yeah.
going to be not just one gene, but multiples, is there a chance that using those powerful genetic analyses, you could identify those traits which give it a characteristic quality independent of terroir?
Yeah, so when do you want me to go back to the
Yeah.
to the question about gene editing? But yeah, I think the new genetics, new technology that's out there will allow us to do quite a lot. But it will take a lot of phenotyping, and the flavor chemistry is very complex. And you combine that with yeast chemistry and the interaction of the yeast with the grape and the environment, getting to varietal typicities and its inheritance will be difficult. But cis-rose oxide is the chemical we focus on for the inheritance of Gewürztraminer-like flavor. It's that particular aromatic compound that we find in Gewürztraminer, we find it in Traminette. I think if you could focus on a particular compound, it's doable. I think genomic selection techniques may eventually enable some of what comes in the future, and there are some researchers working in that area. So if I can go back to gene editing, so it gives me a great opportunity to point out that the VitusGen 1 and 2 projects were followed with a VitusGen 3 project. which includes genome editing. It includes looking at genes that are responsible, MLO genes that might be responsible for susceptibility, and if they are knocked out, can we make a vine resistant by knocking out certain genes? So there's work along those lines taking place in Vitis Gen 3. Vitis Gen 1 and 2 are now history. Vitis Gen 3 is underway for the last couple of years. They are focusing on gene cloning for powdery mildew resistance genes, focusing on gene editing and a variety of other phenotyping robotic technologies as well. Yeah. You want to add anything?
Yeah, I just can't believe that we can't do better than something that came about in the 1700s, right?
Right.
I mean, think if we'd stopped breeding corn in the early 1900s, you know, we would be in a different place. So I find it hard to believe we can't come up with something that's equal or better than Cabernet Sauvignon. Maybe I'm wrong about that, but the other thing I wanted to mention is Andy Walker's 5 new varieties that he produced in a series of back crosses in about 10 years, wasn't it? 10, 15 years?
2-year cycles.
Yeah.
Across.
Are
Two-year
any
cycles.
of these hybrid varieties on the market yet? And how will we be able to recognize them if we wanted to embrace hybrids for sustainability reasons?
Some of them are named varietally, and some of them are named with fanciful names like Heart of the Lake. So I think as Bruce's, as Aravel gets more commonly known, I think people are going to highlight the fact that it's Aravel and put that on the label instead of what people called it New York 81. So there's no guarantee that you're going to find these things in wine stores, but certainly at the local wineries, you can always ask people, you know, do you have any good hybrids?
Put it on your website.
Yeah. And we'd love to stop calling them hybrids. I mean, we have hybrid strawberries, we have, you know, but nobody calls strawberries a hybrid, you know, and it's just the way, it's just the historical thing that came up because of the phylloxera epidemic and what the French did. But it's such a, you know, sort of derogatory term now that, well, why don't we call them the locally adapted varieties, the, you know, the non-invasive grape species, because in a sense, Vitis vinifera is an invasive species too, sort of.